MS translators. So I'm here today, sans Brett. He's busy recording his podcast right now. So I'm filling in, and I'm going to be talking to Dr. Alban Gaultier today. Alban is a professor of neuroscience at the University of Virginia in the Brain Immunology and Glia Center. And he's joined us here at the ECTRIMS conference. And we're just going to talk a little bit today about what we've experienced here at the conference. And also I want to talk to Alban about some of his work and MS in the field in general. So Alban, why don't you go ahead and tell the MStranslate audience a little bit more about yourself and your work that you do at UVA.
Hello, yes, I have a lab at the University of Virginia, and so in my lab we are looking for new therapeutics for MS and of course other neurodegenerative disease. And my lab has two topics of interest. The first one is finding way to repair myelin to the brain, and then the second one is to understand the gut-brain axis, studying the microbiome and how it can influence immune system response.
And so, just as a full disclosure, Alban and I go way back. Alban is actually responsible for me being a scientist. He basically taught me everything I know. Alban was a project scientist at UC San Diego when I was there as a graduate student, and I was not a good graduate student until Alban got a hold of me. And Alban's actually a co-inventor on several of Novoron's pieces of intellectual property, and he's been a collaborator of Novoron's for quite a while. So, Alban, I know a lot of the story, but I think it'd probably be interesting for the MStranslate audience for you to give them a little bit of background on why you're interested in MS.
Oh, that's a good question. It started as a scientific curiosity or lack of being able to choose. I was always excited by immune response, immunity, and neuroscience, and I could not pick the topic that my lab will study. So I found a disease that combine both, and that's multiple sclerosis. And then, I got into it by studying a protein called LRP1 that Travis also worked on, and so we made some very interesting discovery about the function of LRP1 in microglia, which are the immune cells of the brain, and also in oligodendrocyte precursors, which are responsible to repair the myelin.
Yeah, so Alban's work really has been kind of seminal in characterizing the role of LRP1 in both the microglia and the OPCs, and the roles are quite right. Different, and it's actually quite fascinating because it's really difficult to simplify any single role for LRP1, but it does seem to be pretty clear that in microglia it's a good thing, and OPCs maybe not so good at least in terms of MS. But I think one of the things I've talked about in the past with Brett is just going back to your original blood paper, where you basically just were the first person to even characterize myelin debris interacting with LRP1 in general. And I remember meeting Steve for the first time and kind of probing him, and he seemed almost completely naive to what that might mean. I don't think I've ever asked you the question. Did you know what you might be onto when you published that blood paper?
No, it was not a blood paper. You're talking about the G-cell science, where LRP was shown to be a receptor for myelin debris. Mm-hmm. And we came at this with a very narrow monodimensional vision that LRP1 is a scavenger receptor and that it can clean debris. And so the question was, can it clean myelin debris? And of course it does, but it opened a full spectrum of biology that probably also put me into that field. Yeah.
So where did you even come up with the idea to look at that? I mean, because you guys were a cancer inflammation lab, what even got you to think about myelin and LRP1?
I think the fact that myelin is basically a high content of fat, and that LRP has been involved in clearance of LDL particle and other fat product was logical. And I didn't come up with this idea. It was a project started by someone else in the lab that they did not finish, so I kind of picked it up. His name was, he was a scientist. I can't remember his name.
Well, I've never heard of this person, so this is news to me.
Yeah.
Breaking news.
He's a co-first on that paper.
Interesting.
He didn't stay very long, but he started actually looking at that question. Can LRP1 clear myelin debris? And he didn't complete the work, but I did. And then I picked up and moved on. That's the way science goes. Sometimes it's an accident, and sometimes it's lucky accident.
It's crazy to me how that single paper that was so simple, LRP is a receptor for myelin debris clearance, has now turned into this, incredible kind of like diverse spectrum of work that's now spawned an entire company and, a lot of other stuff. So now in terms of like where your lab has gone now, you've gone beyond MS quite a bit. I think the audience would be interested to hear kind of some of the other interesting stuff that you've been working on. Also, obviously MS is a big focus for this, but I think that one of the things that I've always been admired about you and your work is that your curiosity has always kind of been unbridled in the sense of you went where the science justified, not necessarily where it made the most sense in terms of publication. So go ahead and just tell us some of the other cool things you got going on.
Right. So the main idea of my lab again is to find new treatment for MS patients. That was the foundation of the philosophy of my lab, right? So we have this cool work about OPC and how we are trying to make them do more myelin, and I can come back to this, right? So this is a question that many groups are following, but I think our unique approach is, unlike the rest of the world that think that OPC are only doing one task, which is to make mature oligodendrocytes, we actually believe that OPC are very multifunctional cells that can do a lot of other function, and that we think that in the context of MS, they get told by the environment to do something else, whereas they can either contribute to the immune response, they can open the BBB, the blood-brain barrier, they can make cytokines. So we think in MS pathology, OPC is being reprogrammed to do something else. And if we understand that transition and put them back on the right track, we think that we will make them do more myelin, and that's our unique approach.
And just to recap, because I know we've talked about this a lot in the past, but the OPCs are very important in both Alban and my work in terms of they're the progenitor oligodendrocytes. They're the cells that if we can coax them in the right direction, they become the new oligodendrocytes that will repair the myelin that was lost in whatever immune attack was taking place. So obviously we're here at ECTRIMS. This is, I believe, your first ECTRIMS.
My first ECTRIMS, yeah.
And this is my second. I've done some of the virtual ones, but this is only my second time being here in person. So real quick, without going through everything that you saw, actually, I'll focus in because I'm curious, and we're obviously both interested in the remyelination component, but we happen to sit in on the same remyelination section where they were talking about imaging endpoints. Because for me, I think in any disease, one of the big limitations is if your drug's going to work, you need something robust that shows it. You need some sort of test or measurement that says it was a here, then it's a here, and that's statistically different. And so if you don't have a good test, that can be really problematic. And in terms of remyelination, we just want to see the myelin repair. And in a lot of ways, MS clinical trials have been based on behavioral outcomes, and we don't really know a clear understanding of what the behavioral what the behavioral outcomes will be of remyelination. But more importantly, we have no idea how long it might take before those behavioral benefits are seen, and so we're really limited in trying to see imaging as an endpoint in remyelination. And I thought that there was some interesting things that came up in terms of imaging and remyelination, and I'm interested in your thoughts in that section.
All right, so you're correct. Without the capacity to see remyelination happen with proper clinical imaging, all those clinical trials are incomplete because we only assess motor function or optic nerve function, but we don't know what's happening in the brain and if myelin is repaired, right? And so this is the main problem for the field, and we've seen some progress. There's a couple of techniques that were presented that seems to be able to detect remyelination, so it is helpful, but we are talking of state-of-the-art imaging in advanced research facility, and for this to become a standard operating procedure in a clinic, there's still a lot of work to do, right? So progress, perhaps not fast enough, to me. I'm not a very patient person.
I think I share in that concern because one, we're still just scratching the surface. The consensus hasn't really been built yet. I mean, we were just talking a minute ago about how, while they were presenting a very conservative outlook on some of these measures, other presenters in sessions unrelated to the remyelination session were talking about similar protocols, and they were discussing them as though, almost as if they were a given, as if they were accepted.
Right. We all tend to make the best of the data we have, right? And I think another big issue, which is also massive limitation, is we know that the lesions are not only limited to white matter, which is the place where most myelin is. There's plenty of lesions in the gray matter, and those ones are still terra incognita. It's uncharted territory.
Yep.
We don't have a way to see them, and they are as important as a lesion in white matter. And so at best, we can see some remyelination in white matter, and in the gray matter, it's still undone. So a lot of work to do, a lot of people looking for biomarkers, and it will come, but it's a bottleneck for drug development. Yes.
So I would, almost taking, I'm curious about both your opinion of the remyelination information we've gained here, but also taking a step back outside of here, like, what's your general feel on the state of remyelination? Did you hear anything in your time here at ECTRIMS that was encouraging, discouraging, or new, or do you have a perspective outside of ECTRIMS that makes you feel optimistic or pessimistic?
I think one of the most telling talk was by Dr. Lubetsky, right, where she presented what is the status of clinical trial for remyelination, and the take-home message, unfortunately, was not amazing. There's maybe one drug that seems to be doing something, which is clemastine. The rest has been failure. Let's be honest, right? And that's why we need more researchers to come into that field, perhaps from other field, right? And also change the dogma about that idea that the oligo precursor only have one function, to become oligodendrocyte. And I think it will come. The field will slowly change. But yeah, it was kind of, to me, it was old school thinking. And hopefully it will change, yeah.
I mean, I would echo that. I thought that some of the interpretation and presentation of the trials that have taken place was a little bit oversimplistic in terms of, like, for example, they didn't discuss the differences between the Renew and Synergy trials for the anti-lingo antibody, which is a pretty profound distinction, if you ask me. And then, the clemastine stuff has been promising, but it's been slow. Right. And I think that most people have been thinking that there might be a better version that might work. And I think, to me, a lot of the positives that we've seen with clemastine and even with the anti-lingo have been lessons that we can build on. It might not be the drug ready yet, but I thought that the pessimistic kind of overshadowing message or I guess the pessimistic message overshadowed the positives that I do think can be gained and learned from. And that was interesting to me, because that's not an approach that I've seen used often. Is there seemed to be a real emphasis to almost rush to the clemastine. And let's be honest, clemistine's the most promising and it's the one that hasn't failed yet.
Yeah.
But I don't think anybody thinks that clemistine's the best version of what we can do there.
And remember, the clinical trial for clemastine was done not on MS, it was done on chronic optic neuritis, right? Which is a very special part of MS, which is not representing the full disease, right? So I'm really excited to see when they do this with more complex MS population for relapsing or remitting MS, and of course for progressive MS.
Yep.
So yes, repairing the optic nerve is nice, it's encouraging, but it's time to move on to the full CNS and see if it's worked there too, right?
Yeah.
And then there's always this dichotomy, or it's not a dichotomy, but remyelination is often, you have to separate inflammation from remyelination, right? And so people say if we turn off inflammation, we can repair, but we don't really know that because of the limitation of imaging technique. And that's something that often scientists put together as a selling argument, but they don't have the right answer.
I actually thought it was super interesting when the woman from the NIH stood up and I think she made a similar comment. She mentioned, we really only have one thing that actually eliminates the neuroinflammation. So making those comments about how much we might be able to turn up or turn down the inflammation as a precursor to remyelination seemed to be a bit of an overstatement, because short of HSCT, we really can't turn it off. So it's almost like if you really want to be successful short of a real aggressive cell-based therapy, you kind of have to be able to promote the remyelination in the presence of inflammation.
I mean, we always say this, it's a two-phase attack, right? You have dampen inflammation, repair the myelin before the neurons are lost. That's the only thing that matters, right? Because if you're too late, the neurons are gone, and there's no point of repairing the myelin.
Yeah, I agree. Yeah. And it's interesting because going back to the OPC versus microglia comment. So we just had to edit out a little technical difficulty there. But getting back to we were discussing the OPCs, and it's actually been interesting because I think I've seen a lot of progress in terms of people's understanding of like the multiple populations and different roles for microglia, which is something that you have been kind of a pioneer in, especially in terms of the LRP1. And the interesting thing to me is we've actually been playing with the idea of these OPCs, and OPCs have been known for their central role in remyelination for a long time, but we still seem to be stuck on this myopic perception of OPCs when really OPCs are this really diverse multifunctional glial cell that, yes, they are a precursor, but they are not just sitting there as a satellite cell waiting to do things. They are very active.
Yep.
What do you think is the kind of where do you think is different between the microglia community and the OPC community that has made this like embrace of this more complex, nuanced picture in microglia, but we're still trying to stick to this oversimplistic view of OPCs?
Oh, that's a good question. I think it's multifactorial, right? First, microglia research is ahead of OPC research, right? People have been working on this for more time, and they knew that they were multifunctional, so we don't have to change that concept, right? So I think microglia research is about 10 years ahead of OPC research, which is one. And two, yes, the problem is with OPCs, if you open any textbook of neuroscience, it's like OPC is there to make more money, right? But the math doesn't make sense because we have way more OPCs than we need to repair myelin. There are as many OPCs and microglia in the brain, 5% of our cells, right? And they constantly divide.
Yep.
They constantly multiply in a cell. And why are we spending so much energy maintaining that cell population if it's only to make a little bit of myelin sometime, right?
Yeah.
And so all the idea are there to support the premise that OPC are actually poorly named. That's what I like to say, right? We call them oligodendrocyte precursor cells, but this is the worst name possible for them. And I think where the field is going to go is we're going to separate those in subpopulation, and one of them will be OPCs, and the rest will be something else.
Interesting.
So perhaps incoming is.
Interesting. So I'm wondering two things. One, what are you hopeful for now that we have hit a new swing for both your lab, and what are you the most hopeful for in the next 12 to 18 months for the MS field in terms of what you think the next kind of a cool thing coming on the horizon?
Yeah, I want to see more. I want to see more clinical trial focused on myelin repair. We have enough anti-inflammatory drugs. I think that we're at 30 now on the market. They all work great. They all come with pros and cons, and I'm not going to talk about this. I think the market is saturated with anti-inflammatory drugs, but we need to tackle the next question, which is the one that matters to me and to Travis too, which is we need to repair the myelin. Because MS is a neurodegenerative disease, and without proper repair, we can lose the axon, we can lose the neuron. So I hope that we're gonna get in the next 3-4 years more clinical trial to tackle that question. And now that the lab are up to speed and are starting to embrace OPC diversity, maybe we'll come with different type of thinking, right? Can we tell a pool of multifunctional cells to mainly focus on myelin? Or can we take them from an inflammatory status back to a pro-repair status?
Mm-hmm.
And that's what my lab and other labs are starting to explore, and I think that holds a lot of promise. And then lastly, another option is maybe we can try to protect the oligo before they die, right? This is also something very cool. Oligo are a beautiful cell that take care of many neurons into the brain, and they get stressed, very much so in MS. And if we can find a way to unload the burden on them, we could save them. I think we saw very good talk on this. I was trying to make oligo do less of ion buffering, which was very neat. Like actually, we talked about this.
Yep.
It was a cool talk. Yes.
Yeah, I'll be honest with you. In my experience, oligodendrocytes are just the most fragile cells in the world. So anything we can do to prop them up, they more than any other cell I've kind of worked with or dealt with, like they seem to, if you look at them wrong and get offended, they're going to die. So it's a low bar.
They have a lot of jobs to do in the brain, right?
They do.
They have to
they do.
make the myelin, but also supply them all the goodies to the neuron, right? Which are very finicky too.
So. And I think it's important. I mean, like it's a basic concept, but I think it's really important to remember, like we have myelinating cells in the brain and we have myelinating cells in the body, and the myelinating cells in the body basically will myelinate one axon and do that one thing. Right. But the ones in the brain will myelinate hundreds of axons, and they'll be providing different levels of care and insulation to 100 different axons. I think part of the reason they're so sensitive is because their job is so intricate. But man, do they just like when Schwann cells and the peripheral myelinating cells, if they get angry, they kind of just retreat, divide, make a little army, and then fight whatever made them mad. Oligodendrocytes just give up the ghost and kind of.
They have more than one function, right? So the reason why one oligodendrocyte contact many neurons into the brain is actually to synchronize the electrical signaling, right? So they have this very amazing capacity to change their myelination to make all that signal integrate better. And that's also one exciting aspect of research that was not discussed here because it's not the topic, but oligo are able to influence memory formation by changing their myelination, and that's why they're so complex and delicate. Yes.
Yep. Well, that's great. Thank you so much. We appreciate your time. Thank you, MS translators, for taking the time to watch us today, and we'll be back soon with more content here from ECTRIMS.
Thank you, guys.